Okay, good afternoon everyone. Welcome to this course on biotechnology, law and policy in India and the international environment. I'm sure you've gone through the brief about the course which is available on GIAN's website. But just as a matter of introduction, the course is dealing with the developments in the area of biotechnology and how those developments create opportunities and pose challenges from policy and legal perspective. That is basically the underlying purpose of this course. And for this course, we have two professors from Australia visiting us, fairly well known in the area of biotechnology, law and policy. We have Professor Mark Perry from the University of New England, and we have Professor Preeti Krishna from the University of Western Sydney. So I'll just give a brief introduction about our guests, and then I'll invite Professor Perry to initiate the course. So Professor Mark Perry, as I said, comes from the University of New England, Australia. His research focuses on the nexus of technology and law. He has taught around the world, including Japan, New Zealand, Canada, India, and Australia. And by the way, he has visited NALSAR earlier also, but I don't think at that time you were there. I was surely there as a student, so I have attended his lectures. Professor Perry has published over 60 peer-reviewed papers in both law and technology journals, and he is the editor of a recent book, Global Governance of Intellectual Property in the 21st Century, published in 2016. Recently, he was involved in Genome Canada project researching on the ethical, economic, environmental, legal and societal aspects of utilizing genomics approach to pest management. He is directing the project Seed Banks and Loss of Inherent Value in Seed Banks, that is examining the management of traditional knowledge in the process of collection of seeds. Professor Preeti Krishna holds the Vincent Fairfax Chair in Sustainable Agriculture at the School of Science and Health in the University of Western Sydney. Before this, she was also in Canada. She had a successful career of 20 years at Western University, London, Ontario, Canada. She has a master's in microbiology, so we have a scientist and a law professor with us. And she has a PhD in biochemistry and post-doctoral training and independent research career in plant molecular biology. Professor Krishna has applied her diverse research background to developing pioneer projects and implemented an integrated and collaborative approach to her research together with rapid adoption of relevant new technologies and approaches. Now I have a long list of what all she's done in her research, but I'm sure if I read that, very few of us will be able to comprehend what those areas are. And in fact, Professor Preeti's presence here is, particularly to help us understand the science part of, of the whole subject matter, so that we are better equipped to then understand the law and policy aspects of it. So that is brief about our guests. Now in terms of our lecture schemes, there is a slight modification. So today, instead of having a 3-hour session, we are closing at 4:30. So we have a 2-hour session, and then tomorrow we will have a little longer session. So today, basically, Professor Mark Perry will explain the nature of the course and what all we are going to do in the course, and whatever introductory information needs to be conveyed to you, that will be done by Professor Perry, and then Professor Preeti will help you get comfortable with what is happening in the area of biotechnology, what the terms which get used and how do we understand them as non-technical people, so that when we come across those terms in the area of law and policy, we are better placed to have our view vis-a-vis the same. So with this brief introduction, I think I'll invite Professor Mark to speak to the students.
Thanks,
So this camera doesn't work that side, so you have to stay here or
I
this
can't
side.
do that.
Yeah,
I won't see that. Sorry, here. Perhaps it's better this way. So, good afternoon, everyone, and it's my pleasure to be here under this GIAN project, and I must first of all thank Saurabh and also Professor Danda as well for their efforts to make sure that this course came about. So today really I'm going to start off really talking about what the course is about and why it's important for you as lawyers. We'll talk about some of the issues briefly, and many of these, of course, we will revisit over the next few days. Professor Krishna will introduce to you some of the basic science that will help you understand how the law interferes or doesn't interfere with what scientists are doing. Without understanding what the science is doing, it's very difficult for regulators to know what we should be doing in terms of supporting or restraining various scientific activities. Now, in terms of questions, I put it questions at the end here, but I am more than delighted to entertain any questions regarding the law as we go through this course. So at any time that you either can't hear what I'm saying or don't understand a word that I'm saying, then please do interrupt and ask for clarification, because I'm not psychic, well, I'm not in India, and I don't know if you understand unless you tell me that you don't understand. So I must insist that you do ask questions as we're going along, and so please do that. The scientific questions ask Dr. Krishna rather than me. So we'll start off with introductions, but these have pretty much been covered by Professor Bharti. And I think you're quite lucky to have the three of us as a combination here, a trifecta of international approaches to legal regulation in biotech, a leading molecular biologist across the world, and also someone who can give you deep insights to what's happening here in India. Just to give you a bit of context, and context is always good to help people understand where things are and what happens where. Australia, I guess you know where that is. It's sort of south of here and a bit to the east. And Professor Krishna is from Western Sydney University at its Richmond campus, which is in the northwest of Sydney. I am from the University of New England, which is a rural university surrounded by fields, trees, sheep and cows, and some crops, which is also in Australia there, but a little further up. To give you some idea of scale, to fly from Armidale to Sydney, that little gap on the map there, takes about 1 hour. You have to remember that Australia is a very large island or small continent, depending on how you think of it. I guess you all know where Professor Bharti's from. He's from here. But just to give you some idea, if you were to fly direct from here to Sydney, it would take you about 15 hours. We don't want that. So, please now think about what we are doing over the next week. Really, what we're talking about is how biotechnology is being used to create a better environment for animals, people, and crops. This, of course, involves interventions in terms of biotechnology, and I give biotechnology a very broad definition, and I'll talk more about that later. It requires interventions using biology, applying biology, which is what we call biotechnology, to alter how natural systems behave. In some senses, this can be quite controversial. Indeed, in some nations, if you talk about altering biology, people get quite upset and they fall into polemic arguments of why this is either evil, anti-godlike, or a problem. However, as humans, over the last many thousands of years, this is exactly what we have been doing in order to try and make nature fit our needs. Many people will say you shouldn't interfere with nature, but of course this is exactly what we do all of the time in order to survive. Anyway, we'll come more to that later. So first of all, let me just talk a little bit about how this course is being laid out for you. And really, it's stepping stones from today into tomorrow. Today we're going to talk about some of the concepts, the legal concepts, the issues that arise in our society due to the introduction of modern biotechnology techniques. And it's mostly the modern ones that people have concerns with, not the more ancient ones. Tomorrow we'll look at biotechnology and intellectual property, and some of the global approaches through transnational, international agreements as to what's been done on that front. And also we'll have a look at some of the Indian perspectives on intellectual property in this field. And then on day 3, as you can see there, we'll look about why biotechnology and also regulatory challenges. And day 4, we'll look more at regulation in areas such as cloning and in human reproduction. And then day 5, we'll finish up with bioprospecting, traditional knowledge, and also the balance between regulation and innovation and how to achieve it. At the end there, you'll see we have an assessment item, which you'll all be pleased to hear is an exam, which means that you need to focus on what is being said during this week and hopefully read some of the recommended papers, because anything that's mentioned in here or referred to in here, you may possibly be examined on. So best to pay attention, and hopefully you will be able to master your exam with no problem. The other important feature which I want to emphasize to you is that in my mind, legal courses are not simply a matter of me reading out what the law is. Any idiot with reading ability can find out what the law is these days. You just look it up. And I tell my students in Australia that in fact law is a very, very easy subject to study. When I did law, of course, it was much harder. We used to have to beat ourselves with sticks and so forth. I'm exaggerating, but then actually finding the legal information was very difficult. In the mid-70s, there were no photocopiers, let alone internet or legal databases. If you wanted to find out the law, you had to go to a law library, and you would have to look up the information you required. Even finding where to look it up was an art in itself. And then you had to read it and make notes in longhand on a piece of paper. And anybody who's seen my writing will know why I had such a challenge in law school, because I can't read my own writing. Luckily, I could remember things quite well. So today, I think law courses are something a bit different. They bring to you complexity, the complexity of our societies, and the way that we need to understand how we manage our societies through legal systems, how we regulate human action through legal systems, and how hopefully we move forward to develop better societies through our legal systems. And this really is my aim, and my colleagues' aims in this course is for you to understand the complexity of biological systems and how we use them in our society, and how they are or aren't or should be or shouldn't be regulated by our various legal systems. So if you can understand that complexity at the end of this week, you're doing well, because I'm sure I don't understand it, but at least you can have a window into it to see what's going on and hopefully draw your own opinions as to what we should or shouldn't be doing in the future. So that really is the course layout over the next few days. So in the next half an hour or so, really I just want to introduce some of the problematic areas that we have to deal with. Technology, of course, is one of them, and technology is notoriously unreliable. Hopefully, biotechnology is more reliable than IT technology, which I've been dealing with at least in the last 20, 25 years, both from a research perspective and from a use perspective. So what is law? You should ask yourself. Probably, hopefully you do that at least every day, perhaps once a week, to understand why you are studying law. Why are you studying law? I don't know, has anybody got a good answer in here? I'll pick on somebody in the back row because they're hiding in the back row. Why are you studying law? Person under the clock. That's you. You like the subject. I like eating a lot of food, but I don't study it too much. Hopefully there's a deeper moral imperative that drives you to study law, in that you want to make society better through legal application. I'm just being judgmental, don't worry about it. So what is law? We ask ourselves. There's an Oxford English Dictionary definition, which is as good as any. It's basically rules in a particular jurisdiction that regulate our actions and may be enforced either through state mandated penalties that we call criminal law, or through civil penalties that we call, at least in our jurisdictions, the common law. We try to make sure that people behave in certain ways through the application of our legal systems. Now, it may be no surprise to you that, of course, legal systems tend to support the status quo, which is a Latin phrase that you all understand. I hope otherwise all hands would all be going up. What's the status quo? Yes, it's you. There's no good looking around. Situation as it is. Perfect. So you do all understand the status quo. That's good. The legal systems are typically extremely slow to change, and probably that is a good thing. If, for example, you wanted to enter a country and they kept changing their visa regulations every 2 months, like India does, it's extremely difficult to know what you should be doing in order to get a visa. Legal systems usually take a long time to change, because people who are in business, or indeed civil society as a whole, in everyday life, they need to understand what they should be doing. And if legal systems are changing willy-nilly every other day, then people would not know what they should be doing. Businesses would not have time to change their modus operandi, the way they operate. There are, of course, many different reasons behind legal systems and why they operate the way they do. There's natural law, positive law, realist law, and many other different philosophies that underpin the way our legal systems are. For our purposes for this week, I suppose it's enough to understand that although the science is changing very rapidly, our legal systems still tend to react quite slowly, so that it's difficult for the law to keep up, or at least manage, the changes that are happening with technology, not just biotechnology, but other technologies too, and I'll give you a brief example of that in a minute. Science, at least this is my explanation of science here, is a systematic investigation of nature to create knowledge of our universe. The typical science method is that you come up with an explanation for what's going on. You test that explanation and see if it really does, in experiments, work out to explain what's going on. If it doesn't, then you need to reimagine why things are happening the way they're happening. Science, of course, is seen as speeding up, and indeed I think this is probably the human condition. The more we know, the more that we have the ability to change our natural environment through scientific interference. The problem, I suppose, is that whenever there is progress in terms of disruptive science, whether this is an improvement for our society or not depends on who you talk to. For example, you may recently have heard that Tesla is introducing driverless trucks, probably in the next 5 years, which will be electric powered. Electric powered is good, much reduction in pollution, but of course if you're a truck driver and you're out of a job, you see this as not a good scientific advancement, because you no longer have an income to feed your family. So of course disruptive science endlessly causes issues for our society. Sometimes we try and manage these issues through legal system, sometimes we just let the market, so-called, take care of what's going on and allow people to manage it in their own lives. Look at the introduction of cell phones, for example. There is very little regulation on how cell phones are used. I know at least I can see 2 people using cell phones at the moment in this classroom, for example. There's very little regulation on how cell phones are used in our society, although of course they've been a highly disruptive piece of technology introduced over the last 10 years, basically. If you look at recent developments in information technology, although the idea of the computer was basically designed in the 1830s by Charles Babbage, the ability to apply those design features and code even that was written at that time by Ada inside a machine that would work was very difficult, because there were only mechanical calculating processes. It wasn't until the early 1940s that the first real computer was built with Colossus in Bletchley Park, and it was used to crack German codes used by the German military at that time. Of course, since 2000, there's been a massive acceleration in the impact on information technology in our lives. If you think about it, it wasn't that long ago that the first iPhone was introduced, a very smart smartphone design, and that's only 10 years old. These things have had a massive impact on how our lives are in some senses easier, but in some senses more complicated. And in terms of regulation, that has been a large problem for regulators. We see now for example, in many jurisdictions that the state, i.e. the government, is introducing laws that allows the state to monitor our communications without search warrant under the name of preventing terrorism. Not just India, but also in places like Australia, internet service providers have to record and keep the telecommunication header data from all communications that go through their networks. Of course, although we're not talking about it this week, it's worth keeping in mind that biotech is not the only technology that is bringing challenges to our society at the moment. IT is also doing that on a number of levels, whether it's privacy, how we deal with things like ownership of materials which are expressed by people that we call under the copyright of a copyright heading, things like hacking, domain name squatting, data mining, huge databases on your private information, and so forth. The rapid increase in developments in biotech are not behind those in information technology at all. It's probably they're just not so obvious to the general consumer. All of the IT progress has been very, very rapid, but it's also been in the user, consumer's face. The IT companies have managed to monetize and popularize the application of information technology into the marketplace very effectively and very rapidly. This is not the case with biotech. It is much more hidden what has been going on, not so much because people have been trying to hide it, but simply because you just don't notice what's going on, and it's not something that an individual user will be accessing, although perhaps that will change with direct individual medicine, gene-targeted medicine in the future. So you can see here that although things like genetics were talked about and discovered by Mendel in the 1890s, it's probably been only in the last 40 years that we have had the ability to manipulate directly. Of course, we've been manipulating indirectly for many, many eons, but manipulate directly the coding for life forms. So that really is our focus today. And for the rest of this week, we're going to look at animals, we're going to look at plants, and we're going to look at what is happening at the science level that's going to influence us as a society. So what is biotechnology? Simply, it's applied biology. There are some traditional definitions here. One I've taken out of the United Nations Convention on Biological Diversity. If you haven't read that already, you should probably have a look at that. It's a great convention which is positive in its outlook on how we should manage biological instances around the globe, that we should share with knowledge is the basis of that convention. So it's worth having a look at that. It's quite a nice piece of international cooperation. It's probably not given enough strength, but we'll talk about that later. From a standard biotech textbook, it says using living organisms or their products for human benefit to make a product or solve a problem. Again, this is a very optimistic approach because, of course, many instances of the use of biotechnology has been not for human benefit, but for human detriment. You think of biological weapons, for example, or even chemical weapons. And of course you can see they're not for human benefit at all. But at least it's trying to be optimistic, which is good. Biotechnology is not new at all. So you see there's a clay pot there which was dug up about 5 years ago, and it was investigated, and it was found that there was an application of biotechnology for a particular purpose. And I wonder if any of you can guess what it is, or know what it is. What were the Chinese using biotech for in a clay pot 9,000 years ago? I have to pick on someone if no one volunteers. Sorry? No? Long shot guess? It's good to have a guess. This is good on you for that. Anyone else? Silk? No, that's a good guess too, but wrong. Tea? Tea. Well, you're getting warm. Anyone else? You have to shout loud because of the paper. They were good on paper, but that wasn't what it was used for. And that, cocoa. Chocolate, that would have been a nice idea, but I think they were a bit more hardcore than that. one more, take one more, really good. Opium. Also warm, but it was actually the British that introduced opium to China. Exactly, alcohol production. Chocolate fish for that man. So the Chinese were actually using enzymes and yeasts 9,000 years ago to produce alcohol, and there's direct evidence for that. not just accidental production of alcohol, which happens in nature, of course. If you just leave some apples in a jar, for example, you'll end up with alcoholic cider. You don't have to do anything, just leave it in a jar. They were targeting the production of alcohol in their clay pots. And in Egypt, some 4,000 years ago, they were breeding geese, deliberately selecting the best geese to produce more geese. They didn't know genetics, they didn't know how genes worked, but they knew if they bred two big fat geese, they were likely to get another fat goose. So they were using some knowledge about biotech to produce, by ensuring correct couples mated and produced eggs, a particular strain of geese. And of course, here in India, some 3,000 or so years ago, Ayurvedic compounding was very well documented, and you can find that written today. The production of cheeses and yogurts and alcoholic beverages are probably the first instances of the application of biology for human, or at least some people say human benefit. Of course, there are many, many legal problems that arise from these efforts to intervene in natural processes. Sometimes we try to ensure that we encourage innovation by allowing people to patent materials. So what about patenting human genes? Should we be able to patent human genes? This has been a very hard-fought battle within the last 10 years, and I think it's probably come out in the right direction, and again I'll talk about that later. The argument, or the ongoing arguments about so-called genetically modified foods, where we have targeted the introduction of bits of genes from one variety, or indeed even different species, into a food crop. There's the arguments over cloning. So there was a crazy religious group called Clonaid, the Iranians, who had claimed to have cloned human beings in Australia some 15 years ago, but there's no evidence that they actually did, apart from their claims. But should we allow people to clone other people? There's arguments over the use of stem cells. Stem cell therapy is now starting to show some positive results around the world in treating previously intractable diseases. What are the ethical, moral and legal restraints that are placed on the use of stem cells? Biological weapons are banned by a number of international treaties, although some people say that the research into biological weapons, for example the smallpox biological weapons, led to the ability to create new vaccines. And the only reason that that came about was because of the huge amounts of money that various jurisdictions were willing to pour into military questions rather than medical questions, but they came out with a medical answer from a military so-called problem, the problem being how to effectively kill as many people as possible. Sadly, that is the way states often operate. They put a lot of money in military, but happily sometimes we get a positive result even out of those things. Biological data, and biomedical data in particular, has a great potential for use across our societies in order to identify how diseases occur, when they occur, and who are the likely subjects for the disease to occur within. For example, genetic testing can identify now quite a large handful of diseases where malformed genes, will give a very high penetrance for the occurrence of that disease. In other words, if you have the identified malformation in a gene, you're very likely to get that disease. And so on and so forth. This raises, of course, many, many problems. The story of recombinant DNA, so-called genetically modified organisms, has quite a short history. It's only really been since the early 1980s, which may seem like a long time to you, but for me it's not such a long time. It's only since then, in the last, 35 years, that people have been able to directly modify genetic sequences. There have been some remarkable failures on this front. For example, the Flavor Saver tomato that was approved in North America for marketing. It was a tomato that was based on, unfortunately, a rather poor variety in terms of flavor, but they inserted genes in it that allowed it to keep a longer shelf life. Unfortunately, the tomato they resulted with, which they tried to market and was on the market for some years, was pulpy and not very tasty and met with a high consumer rejection. The consumer rejection, it should be noted, in North America was not because it was genetically modified, but simply because the product itself was not good. It was not a good tomato, so nobody wanted it. Other stories, such as the golden rice story, to try and avoid the instances of shortages of vitamin A in a number of developing countries by inserting genes into rice that would make it yellow because it had precursors to vitamin A in it, also has not had much commercial success, mostly because of political resistance. However, some crops have had remarkable success in terms of their influence on the market. If you look in Australia, which has had a huge growth in cotton plantations over the last 40 years, nowadays about 99.5% of all Australian cotton is GM cotton. Most of it is stacked, so about 98% has two genes in it, one to prevent cotton borer weevil and the other to make it Roundup Ready resistant. And now, because of this, because of the harsh Australian environment, they can control weeds easily, and they produce cotton that is now economically viable and is a major export crop. If you look around the world, there's some 185 million hectares. That's probably, I don't know how big India is, I should have looked it up, but I didn't. But that's a large area in terms of arable land, is now being planted with biotech crops of one sort or another. So that's just a little introduction to those issues. But one issue that we need to face in terms of legal research in this area is not just the biology terminology that Professor Krishna is going to look at, but the everyday terminology that we come across in our readings. So, for example, people talk about natural foods in the market. People say, oh, I really like to eat natural foods, not ones that have been interfered with. However, if you look at the history of the development of something like a staple crop such as corn, certainly in North America it's a staple crop, you look at 5 AD, that's 2,000 years ago corn, you'd be lucky if you got half a dozen, 6 or 7 corn kernels on a cob. That on the left there is a typical cob from corn grain grown in that era. Through traditional, so-called traditional breeding techniques, we have isolated and developed high output corns, such as the cob on the right, which you probably eat whenever you go and get a bit of grilled corn at the market. It will not be one of the corn cobs that's really natural on the left, because think of the effort required in order to make a meal out of that corn. You'd have to harvest dozens and dozens of the cobs. Indeed, most of our modern crops have been developed over the last 1,000 years through crossbreeding, interbreeding. Indeed, the basis of corn, wheat rather, that we use in our chapatis, rotis, etc. every day is not really a natural product in terms of, it's not something that nature invented. It has been through careful selection and crossbreeding of something called einkorn and other varieties that produced bigger grains, a better food source, and indeed this type of wheat would not survive very easily in nature. If you are a corn farmer, a grain farmer, a wheat farmer, you have to work very hard to maintain your wheat crop. It is not a very vibrant crop. It's very prone to being invaded by weeds, which are typically stronger, and so forth. Our modern wheat crop is highly selected and has been modified over and over and over the centuries to produce what we call natural wheat today. So what do we mean, of course, by natural modified, genetically modified, is of course an argument that has really been taken over by polemic, by people just ranting about particular directions. You could say that all of our food has been genetically modified because we've interfered in how it has developed ever since we started farming. So I suppose the argument today is that developments that we are using nowadays are perhaps more extreme than the ones we used 200 years ago, simply because we have the tools to interfere in different ways. And indeed, people say if you put a gene from something else inside a tomato, then it's no longer a tomato and it's an unnatural food. So these are the arguments you have to think about, and in some jurisdictions they actually legislate based on those arguments. In fact, most jurisdictions have some kind of restriction on new varieties, either because they're simply new, such as in Canada, it's a variety that has not been produced before. It doesn't matter how it's produced, it's just it's a new variety. And those that are not. And indeed most, as I say, most jurisdictions have regulation. However, people tend to get less upset over genetically engineered medicines. So you won't find many diabetics complaining about the fact that they're using humulin, which is actually produced by genetically engineered bacteria to produce insulin that's very much like human insulin, rather than ripping it out of animals and getting rather a poor variety of insulin. So indeed, vaccines, many vaccines that we are happy to accept to protect us from things like hepatitis B, Ebola or SARS, have been developed using genetic engineering. So the way we approach how these things have been developed is highly biased by our own preconceptions of what we think of as being either ethical, moral, or so forth. People take very different approaches depending on their background. If you are of a scientist bent, you think of the alpha male scientist, their solution to a problem is, we will fix it. It's not that we're going to avoid the problem in the first place, it's that we will develop a solution. Often the best solution is actually to avoid the problem. But rather than that, people look at, let's apply science to provide a solution. There are those in politics, for example on the green side of things, that say nature knows best. Mother Nature knows best. Let's leave it to Mother Nature. Of course, if you were in a typhoon or you're bitten by an infected mosquito, or one of these other terrible things that nature provides you, you're probably not such a keen supporter of nature. Others, perhaps the religious right, say life and the construction or development or pointing of life in a particular direction is solely the domain of God, and that humans should not interfere with it. This, of course, is ridiculous, because ever since there have been humans, we have been interfering with the way that nature is dealt with. Of course, as lawyers, we tend to think that regulation can manage it. If there's a problem with some science application, scientific innovation, or a potential problem, certainly from a political standpoint, a potential problem that the populace is likely to get upset about, and you see that in parts of Europe, not all of Europe, in parts of Europe with genetically modified foods, then we need to make a law against it. Of course, a lot of people say there are huge risks with our interference in nature. Of course, there is also the avoidance of risk with the interference of nature. For example, corn, and I talk about corn because it is a major crop, but you could say similar things about rice as well. If you have corn that has been chewed upon by various bugs that like to chew on corn, it tends to leave the corn with an infection that produces mycotoxins. The mycotoxins caused by these fungus will actually destroy your liver over a period of time. If you develop corn that is not palatable to bugs or kills the bugs, then of course you avoid that risk. So you have to balance the risk. So the risk of eating BT modified corn is what, you have to ask yourself, as opposed to the pesticide sprayed, fumicide sprayed, fungicide sprayed corn, or the corn that's not sprayed at all that's likely to have fungus on it that's going to rot your liver. So these kind of balances are not often taken up in the legal debate in terms of regulation of GM crops, or indeed even in the application of things like fungicides and so forth. The balance of risk seems to have been left out of a great deal of the discussion. To get back to our discussion on terminology, if you were to talk to somebody in the Delhi metro and ask them if they knew any of these terms, they would probably be aware of things like cloning, genetic testing, GMOs, genetic fingerprinting maybe. They've all seen CSI or one of those stupid American programs. Biometric identifiers, maybe yes, maybe no. If you've been to the US, you'll have your fingerprints taken, so that's a biometric identifier, you'd be aware of that. Xenotransplant organ replacement, anybody aware of that at all? No, everybody's, well, no, I might talk about that during the week if we have time then. Chimeras, anybody know what a chimera is? No? So a chimera is where you cross two species. A mule example is an example of a chimera. It's a cross between a horse and a donkey. Natural, though. so not the chimeras that anybody worries about. Sex selection, there are indeed, in application in the US now, the ability to, under certain circumstances, it's regulated, to select the sex of your child before fertilization. And of course, vaccines, drugs, and so forth, everybody knows about. So each of these areas has a contentious aspect. And most of these contentious aspects are due to different moral, legal, and scientific perspectives on what is happening. Most of biotechnology, when people become aware of it, will upset somebody at some point. Indeed, the courts have been somewhat influenced in recent years. Indeed, over the last 7 years, there has been a resurgence on the, if you like, the downplaying of the ability to people to patent genes. For about 30 years, it was the case that genes, whether human genes or plant or animal genes or bacterial genes, could be patented, certainly in the US and in other countries as well. So a battle began in the US, really, and in Europe over the company Myriad, which had acquired patents on the BRCA1 and BRCA2 gene, I suppose you call it deformity in the genes. These two genes, when they were in such a state, have a very, very high penetrance for the incidence of breast and ovarian cancer. So Myriad was making a great deal of money out of promoting the fact that they could test for the presence of these gene instances, and thus could predict that you were highly likely to contract one of these cancers. The gene patents were challenged. They were struck down first in the US and then it was upheld in the US, but then the Supreme Court recently, in terms of legal systems, in 2013 struck down the patent. A similar story with the same gene set in the same company, Myriad Genetics, was followed in Australia with, first of all, the courts striking it down, the appeal court upholding, and then the highest court, which in the Australian parlance is called the High Court of Australia, struck down the genes last year. This has been a major change in approach across the world in terms of the ability to patent genes. It is now hugely difficult to get a gene patent as such. You can, of course, apply for patents on methods for applications of genes and so forth, and we'll talk probably a bit more about that tomorrow. In India, there was a great deal of debate over the introduction of brinjal, which of course the English name is aubergine, GM brinjal. Most of the debate seemed to focus on that this was unnatural, it's going to kill you because it's a GM brinjal or GM aubergine. In fact, I think the major problem with these types of crops is not the actual science or the consumption of the materials in the products, but rather the monocrop problem, where if you have a highly successful and profitable crop of one variety, a GM variety, then you're going to end up with people just growing that variety, and you lose diversity, and you lose, certainly in the marketplace, the ability to buy many of those hundreds of different varieties that are available today. So this was in 2013. So these are indeed hot topics. Wherever you go in the world, you'll find a hot biotech problem in terms of the public appreciation of what's going on. So is this really a good way to manage risk, to manage regulation, to manage perhaps public perception and fear in terms of this new technology? I think perhaps many of the approaches are due to the fact that mega companies are interfering or taking over the production of what is vital to our everyday life. Believe it or not, if you did not have your computer with you, or you did not have your cell phone with you for a week, you would not probably die. However, if you were deprived of foodstuffs or water for a week, you probably would die. So perhaps there is a differentiation in the type of technology here, simply because it's a biological product, i.e. we're talking about something that is essential to our life, whether it is a food product or whether it is direct action on our bodies using biotech materials. I suppose part of the problem is that Hollywood has had lots of dystopic regimes in many of their movies, showing dystopia being the opposite of utopia, showing societies where genetic influence has created either a world of clones or has created the ability of the state to monitor human ability and thus place you in whatever job you think you should be in due to your genetic disposition. There has been, of course, some hysteria in the media. There is inherent uneasiness of people in terms of interfering with biological materials, perhaps one of the reasons being that we really don't understand them, and the ability to gain a rapid benefit individually is difficult to see. There's a great confusion in terminology. Functional creep and definition seepage is a nice term that I've made up in this realm anyway, to show that people don't understand what the words mean, and the words tend to creep into different areas where they shouldn't really apply. I suppose that the influence of large business into such a fundamental aspect of our lives, of course, is uncomfortable for many people. But the monopolies in the tech industry doesn't seem to really worry people, as opposed to monopolies in production of food does worry people, and perhaps that's a good thing. So you have to ask, what are we as lawyers trying to do with biotech regulation? What do we want to do, of course, is maximize overall benefit to our society. What we mean by this is sometimes difficult to determine. Perhaps at least at the minimum standard it should be to produce food where it's needed, rather than overproduce food where it's not needed and then not distributed to where it's needed. Technology does have a role to play in sustainable food production. It cannot be denied. If we understand the complexities of biological systems, at least to a small extent, then at least we can understand somewhat unbiased viewpoint on how, as lawyers or policymakers or regulators, we can manage the innovation that's coming from biotech. We need to balance risks, not just looking at one side of risk, but the whole picture. And of course that becomes extremely difficult because there are so many factors at play. Information technology can help us there because, of course, the newer computers are starting to become rather smart and rapid at managing complex problems. But that's a talk for another day. We want to, of course, avoid hiding different problems behind a smokescreen. Large corporations, of course, that have vested interest will have no incentive to share problems with you. If there is a problem with a developing biotechnology, they won't tell you about it unless they're forced to. Looking for rapid solutions is probably not the answer either. So the regulation has to be carefully constructed, probably not to address a small issue of some developing technology. It needs to have hopefully a technology independent, at least to a certain extent, approach to how we should regulate innovation generally, rather than looking at a simple ideological approach to regulation. So I've gone a little bit over my hour here, but I think probably it'd be a good idea if everybody had a quick 5-minute break before Professor Krishna hops up and bedazzles you with some of the biological science that's going on at the moment. And before you go for tea, and of course the opportunity will arise at the end of the class as well, has anybody got any questions right now? Desperate questions that you can't possibly hold? Nothing at all, it was all very clear. That's amazing, what happened? So let's have a tea break and see you all in, say, 7 minutes. 1:45. Oh, I gave them 7. I told them 7.
And what else do you do? Hi and hello. Anyways, namaste to you all. And it's really nice to be here for the first time in NALSAR and perhaps the second time in Hyderabad. So just sitting here and listening to Professor Perry, I remembered my days in Pantnagar University where I was doing my master's in 1980 when you weren't even born. And I remember that soon after lunch, with the fans going and with this high humidity, there were times when this would happen quite often. So I know it is hard for you all to focus on what is going on here, but today what I really want to do is to just sort of make you aware of the importance of biology. And as Professor Perry said, that there are so many things like your computers and your cell phone, which just goes on right now. You see the value of it. You cannot live lives without it. Perhaps it's your first wife. Your second one is still to come, or the first husband, second one still to come. But the fact is that it's become part of your life very early on. But my question is that this is part of your life. What about your life itself? And the question that I have to ask you is, how many of you really think every day about life itself? How many of you value life? How many of you think about diseases, about hunger, starvation, problems that could happen because of accidents, because of genetics, that can actually hamper life or actually end life? So my question is that how many of you all have come close to any such devastating facts of life? Can you tell me if there's anybody in your family that you have seen suffer from cancer? Raise your hands. Okay. How many of you have seen people been in accidents and become crippled, quadriplegics and so on? Okay. Perhaps you have not seen starvation because India's already overcome it, but how many of you have seen poor people that do not have enough calories? Calories is one thing, but do not have enough as far as a completely balanced nutrition is concerned. Millions, right? Exactly. Now think about it, that how important biology is, how important biotechnology is, and how much we take for granted. Just because you have been immunized when you were a child against some of these diseases, you forget about where those vaccines came from and what's the biology behind it. How many of you are aware, or how many of you scared of the fact that you're not infected by hepatitis B or C or A and so forth? Do you worry about that? Do you know that a person who is infected, I believe it's A or C, I can't remember, but who has even spit in your eyes, you have a chance to get it. Okay? How many of you have worried about ever receiving a blood transfusion and actually thought about what if this was tainted with an HIV virus? Well, people have been infected by simply blood transfusions and have become infected by HIV. I don't know if you think about these things or not, but I do nearly every single day. And I just worry about, and I thank God for the fact that genetically, as much as I know about myself, well, I don't really know except for how I function, that so far I've had a healthy life, and that it's a miracle that all of you who are developmentally sound, just remember if you have seen a developmentally affected child or an adult, what their life holds for them and for the loved ones and the carers, the immediate family who are caring for them. So life is really a miracle, and how we develop into normal human beings is a miracle. And I don't think that we give enough credit to that. And we also don't think about the perils of life that we face every single day, and of the unknown that each one of you doesn't know about. And you really don't know what is there. We think of going to, perhaps asking an astrologer or a palm reader and so forth, that what may the future hold for me. But often you do not think about science, what science holds for you, which is basically what makes you what you are today. So what I want to really, with this introduction, get your attention to understanding the basis of life, and that basis of life is what makes us who we are and what will govern your life in the future. So it is your responsibility that how you will actually look after yourselves, take care of your lives, as well as others around you, and for a larger good cause in the world. And I think to be able to practice law, you must understand the basic biology, because everything is tied to it. Even psychology is tied to it. Not only biotechnology, but even psychology, how the mind works, how we go about, the criminals and so forth, and the forensic science that has come about that catches the criminals and so on. There's so much that is dependent on this technology now, and just generally about biology. So coming to biotech, as Professor Perry has already told you what the definition is, I'm not going to read that out to you, but I just want to again capture your attention to some of the global challenges that we are facing now. I'm not sure if you really understand the impact of climate change. Do you guys understand what climate change is? Raise your hand if you do. So you do know that we are going hotter in this world. Do you realize that we have a lot more carbon dioxide and other greenhouse gas emissions that have polluted our environment? And remember, this is the environment where we have scarcity of water and food, probably in the future, if the population continues to increase like that. And it is in this world that perhaps you will be living, we will be living, you longer than us, some of us, and you will be bringing into this world, at least most of you, if not all, are children. So you have to think about these things. How is it possible to not be interested and to not think about these things? So yes, we have the changing climate that threatens our world, threatens our food security. And then I guess everybody is sort of thinking about where the population will be by 2050. Will be about 9 to 10 billion people. And it is actually said that we need to 2.5 times more food than what we currently are producing. And by that time, we will be consuming 3 times the earth that we live in. Do you understand what that means? Which means that we are already consuming the earth. The capacity of the earth is 1 times. We are consuming it at 1.6 times as it is today. And at that time, in so many years, we'll perhaps be consuming it three times the earth's capacity to be able to keep the population in a good state. So it's not just about living a life that you go through motions and have a house and a car. It's about thinking about these things that we have to ensure for ourselves, that we have quality of health, we have nutritious food, enough food, we have equality in this world, equity and equality, and that we have not only for ourselves but for the entire world. So, just to sort of bring home to you all these, I think that if I can capture your attention with this, then only will I be able to explain to you some of the basics of science, and I hope you will find that interesting because it absolutely consumes me. It consumes me. I don't even know how I can give you an analogy. Perhaps if you've ever, what would be the best way to give? People get addicted to alcohol and drugs. I get addicted to science. Maybe that's the way to put it. So as we move forward in the future, you know that we have problems with water. India's always had problems with water, but now with increasing drought, and we are also using up all our resources in other ways, the soil nutrients and so forth. And then as we keep putting it back, we pollute the environment. The amount of fertilizers we put in, the amount of fertilizers that go in the water and pollute the water, which you drink, and then that causes the problems. What can it lead to? It can lead to diseases, genetic changes, and all sorts of other problems in the future. So let me just say that we do have some major pressing problems in this world, and what we need is, perhaps I will only focus on crops in the little time that I have here, but somewhere we will talk about the medical interventions as well, and we can always talk about it in our discussions. So we need crop yields, we need them improved, because if we don't have them, then of course we may face the problem of food security. Then we need to develop the crop varieties that can resist all the different infections and infestations that might come about because of the climate change. Because these organisms, biological organisms, are changing. We have a lot of genetic changes in us continuously, but some of these changes have no effect on us, some of them have good effects on us. That's how we evolve, and it's selected, those good changes, and a lot of them can also have very negative effects on us. So those are the things which will cause cancer and some diseases and so forth. So we do need, at least talking about crops, we need better varieties where we have better output to input ratio. This only means that if we are consuming so much water and fertilizers, then, which is actually at the moment, a lot of it is being consumed and it's going to waste because the uptake efficiency of the crop varieties is not good. So we have to have genetic changes that need to be brought about. Now I'll give you an example. There was a speaker I recently heard at the Crawford conference, and she's from Zimbabwe, and she was trying to tell us that during her time, she was probably my age or older than me, during her age, her grandmother, she had a little garden at the back of the house, and they had a farm, their own little farm, and on the plate they had a variety of different vegetables, a little bit of meat, which was the source of protein, and so forth, and they had just sufficient amount of nutritious food, and people were healthy and for the most part happy. Yes, they could eat a lot more, They probably didn't have the treats that we have, all the ice creams and the sweeties and all that, but they were healthy and content. In the next generation, they decided that, well, corn is going to give us a bigger income, and so for the greed of money, the next generation, that was the cousin of this woman, he decided he's going to plant just corn. Corn, maize, we don't consume that much in India, but in other countries they do consume corn a lot. And you know what they had as the food, given that they were in a little bit isolated community? She said that the kids basically ate corn for breakfast, corn for lunch and dinner, and the only difference was that the consistency of the food got thicker as it went from the breakfast to the dinner. As a result, the kids were developmentally affected. That's the problem when you have one kind of crop and you feed yourself with one kind of crop. Think of yourself if you were just eating rice. What would happen to you? You wouldn't get enough protein, you wouldn't get enough nutrients, all the micronutrients that you need, the vitamins and so forth, right? So there you are, the children would be developmentally challenged. And as a result, what is needed here, the reason I gave you that example, is that if we look at the bananas that we have globally, not just in India, there's this Cavendish bananas that is, it's actually responsible for 95% of the bananas that are being sold in the world, sold worldwide. But because bananas are seedless, they don't go through fertilization, so there's no exchange of genetic material. They're propagated vegetatively, and so there's this one variety. Now think about it this way. If a pathogen developed for this one variety such that it attacked it and you got no bananas, what would happen? It would be wiped out. So if you have monocrop, one crop, one variety, or few varieties, let's put it, that's not a good way to have your food. The idea is that you have to have diverse varieties so that you can have many of the problems that can be tackled by the diversity in the genetic material. And we'll see what genetic material is in a minute. So what is a genetically modified organism? Everybody in very layman terms, you all understand that it has been developed, an organism that has been developed by making use of recombinant DNA techniques. All right? So the question is, how many of you now understand DNA? Anybody here understands what DNA is? Raise your hand. In schools? Yeah, it's okay. Tell me, how many of you really understand the genetic material? You do? Little bit about it? Not much. Do you understand what genes are? Yeah? I want to hear a definition of a gene from one of you. Anybody. It doesn't matter what you say. You're not scientists, so nobody's going to be judged. You ask me a question in law and I wouldn't be able to answer. I'll say, oh, whatever. Yes.
Genes are protein molecules that contain genetic information. They're stored in almost all the living cells.
So the last part?
They're stored in almost all the living cells that we have.
Excellent. Very good. So yes, it is the material that contains all the information that makes you what you are. And have you ever wondered about the fact that you have 5 fingers and they are all functional? You have 5 toes that are all functional. You have a heart, a lung, whatever. And it's all doing the job that creates life and makes us into what we are. So that's the information that we are talking about, which is basically nothing but gives us life. So what Professor Puri touched on, before I get into the genetics or the real hardcore science, is that why is there so much hype about genetically modified organisms? And what do we need to ensure if we carry out this technology and we actually tamper with the information that we consider as being natural, that we are born with? So if we tamper with it, what are the consequences, or what are the things that we have to worry about? So one of the things is that you have to have a purpose for why you're making a genetic change, right? A question, an idea that, oh, I'm going to improve something, I'm going to change something. And because of wanting that change or wanting to improve, you are going to target something and you're going to make a genetic change. When you make that genetic change in that organism, in the genetically modified organism, the first thing you want to see is that do you get the desired effect with which you intended in the first place. If you get the desired effect, wow, that's a big success. But then the question is that going to actually affect another organism in a negative way, or even in a positive way for that matter? If it's a positive way, then fine. But you have to test that it is not going to affect any other organism or any other system in which we live in a negative way. So that's the second most important thing. And so you are going to, yes?
Some examples about how that would be an unintended effect on someone.
Yes, so we will actually, I'm sure Professor Perry will discuss this, but just as a way to give you an example of making a GM crop. So if you have a GM crop and now your pollen from that crop is available to go and float in the air and go and cross-pollinate some other crop, closely related crop species. And now you end up introducing that trait in that crop species that you may not perhaps want it. Or if, like for instance, people have claimed that just because you have herbicide-resistant plants or insecticide-resistant plants like, your BT cotton and all that, then you, or insect-resistant, sorry, so that it does not end up affecting the rest of the insect population that are not pathogens, or sorry, not pests on that particular crop. Because you can't change that, you don't want to change that balance. You don't want to affect the biodiversity. If all the insects got killed because of that, that would be a disaster, right? So you only want that targeted organism to be affected. Very good question. So you want to make sure that the ecosystem in which we live in is not upset in any major way. And then finally, you want to quantify the economic and social cost benefits and so on of the biotechnological intervention. So those are the kind of things that you are probably more along your alley than mine, but definitely to ask the question that why do we want to make the genetic change? How do we go about making the genetic change? And how do we test the genetically modified organism is pretty much within my domain of what we do, or at least my understanding. So what I have put together is really very simplified form for you, but I think that if you understand, if you just take the time to understand it, it will start making, a lot of it will start making sense to you and you will start appreciating it. So here we are as a human, as you can see, and each of our cells, human cells, you can see that it has a nucleus. And the nucleus contains the DNA. This is the genetic material, which is what makes us into what we are, makes us into a man or a woman, makes us into a healthy human being or a diseased human being, makes us one with genetic defects. If one is born with, 4 fingers or no fingers at all versus 5 nice fingers, and so forth. So you can see that this genetic material is organized in the form of what we call chromosomes. And I'm sure everybody has heard the term chromosomes, right? Yeah? Okay, so we carry chromosomes in each cell, and if we open up, this is just very tightly packed, think of it as though you've tightly packed up a rope into a little bundle. That's a chromosome, but if you open up that rope or a thread, then it's going to be meters and meters long, this DNA material, and this is ultimately what carries the genes that we just talked about, and then I'll hopefully give you a better idea about what genes really means. Okay, so now that you've understood this part, I hope, you can ask the question that if every cell of our body has the same genetic material, then why do we have some organs that have developed into a lung or lungs, and one organ has developed into a heart? I'll come to that question later on, but this is the kind of questions I want you to start thinking of, that if you have the same information, then why is that information making the different parts of our body? Okay? So coming to genes now, you all are living in a time when there has been a revolution in molecular biology, in biology as such, you can say, because our genomes have been sequenced. And what has come out of those gene sequences is the number of genes that we contain. And these are your just a gene is a unit of genetic information, which I will explain in a minute. But just to show you the number of genes that the different organisms have. So you can see that plants, rice and maize have many more genes than humans, but then even the small little worm or the fruit fly, you know that on bananas you will often get that little fly coming around. You can see they're not that far off in their genes, even a small little thing. And the very first picture actually that I had here, this percent is telling you how similar we are to the other organisms as far as our genetic material is concerned. So when the human genome was sequenced, a non-scientist, is asking a scientist holding a banana and saying, you mean, looking disgusted and saying, you mean I'm 50% identical to this banana? And the scientist is saying yes. And that's exactly true. So we are quite closely related to organisms that we do not think about. Okay, so now let us look at, remember that thread or the rope that I talked to you about? That you open up, that's your DNA, that's tightly packed up into chromosomes. Now in that piece of DNA, you will have a number of genes. And what these genes are is nothing but sequences of bases, okay? So that this becomes a little bit technical for you, but just bear with me. So DNA is a chemical component, and chemistry you all understand, right? A little bit. So there are some bases, just take it, just sort of without understanding. Think about it that you have numbers 1, 2, 3, 4. Similarly, we have bases A, C, G, T. And DNA is made up of only 4 bases. That's it. Now what you can do is you can write it either as ACGT, or you can have a sequence AGTA, or you can have a, or you can have CAGG, doesn't matter, the arrangement can be anything. So the sequence just goes on as AACG, TAC, or whatever you want to make it up. But it's just with those 4 bases, all right? So that's really what is encoding the information. The genetic information is just that. And it will become clear to you again that when we have a gene, something tells us where the start of the gene is, where the stop of the end of the gene is. And I'm not going to go into any of those details for you, but just think that interspersed in that thread of sequences are these sequences that we call as genes. So the question is, well, why is it stuck now? Okay, here we are. Okay, so before we go any further, I just want to say that DNA is made up of 2 strands, so 2 threads, and the 2 threads are basically held in this way. So if you look, next to this is 1 thread, 1 top strand. I've only written 4 bases here, but you can go in any sequence that you want in one direction, okay? And then your second thread will be going in this direction, and it is going to be just complementary of the sequence that is on the top. So the only rule that you have to remember is that the A always base pairs with T, and C always base pairs with G, or the opposite way. G base pairs with C, and T base pairs with A, or A base pairs with T, doesn't matter. So now that you have these 2 strands held together by that kind of base pairing, now the second mechanism is to transfer this information of sequence in one of the strands to what we call as a messenger RNA, because DNA cannot be translated directly into protein. So we have to go through this intermediate step, and then the next thing is that this RNA is translated into a protein. Now you're going to ask me, well, why are we going from DNA to the protein? So here is what I would like to say. DNA is the genetic information. Proteins are the workhorses of our cells. You know how we go about doing the jobs, like if you are running, I don't know, a household, each one of us will be doing some job. Well, I guess the proteins are the ones that actually do the work in our cells. They are the ones that give us life. They carry out the biochemical reactions that give us energy, that, let us breathe or digest our food, or, all the things that basically sustain life. So it's proteins that do the work. Now the question is that if you have a defective DNA, you're going to have a defective protein. And if there is a defective protein, the protein cannot do its job. And if the protein can't do its job, the cell cannot work properly. And when the cell doesn't work properly, then our tissues are affected, our organs can be affected depending on what gene that is. So do you see the connection between the DNA and the protein now? Yeah, everyone? Okay, it'll become even clearer to you if you now look at this sequence here. So again, this is just showing you 3 bases tied up in this strand, the thread that I had talked to you about. Just look at the top one and just look at the bottom strand. And you again have that same thing, GC, G base pairs with C, A base pairs with T, and G base pairs with C, okay? Now, when it comes to transferring this genetic information to protein, then if you look, the protein, the amino acid that is coded by this sequence, GAG, is glutamine. Proteins are made up of amino acids. Proteins are also a chain of amino acids, and these amino acids are then folded together to make the protein conformation that you often see protein folded like this. And that's because the proteins have to perform the different functions. So now you see that this GAG, this sequence has now been transferred into the RNA, and the RNA is then translated into an amino acid. Think about it that as we go along and read the sequence in the DNA, you will have 3 bases coding for another amino acid, and then another amino acid, and so forth. So suddenly you have a sequence of amino acids in a protein that is going to make it into a functional protein. Yes? No, they will, you mean all 4 bases? Yeah, so the bases are only 4, but these bases can occur in any combination as you go along. So it can, as I said, it can be ACGT or GAGT, CEC, but I've only written 4 there because I'm only showing you those 4 bases, but the sequence can be any, and it will continue into any sequence that, yeah. Yes, absolutely. But usually you won't have a strand that is missing any bases. There's no hard and fast rule, but it has never happened that a strand is completely missing. You can have sequences in certain regions that will be, for instance, GC rich, and there will be very little AT content there. But if you, because this is a very, very long molecule, you will not end up having a DNA strand that does not have 1 base. You know, that's just how it is. Yeah. Yes?
To convert DNA to RNA is the 5 to 3 strand, the top of the strand is the one taken, or?
Very good question. Oh my God, you guys are getting it. That's really nice. So, yeah, that's a very good question. So if this is the 5' to 3', okay, the mRNA that will be formed, first these 2 strands will separate, and the RNA, the protein that synthesizes the RNA will never synthesize 5' to 3' and say ACGT, because the whole concept of base pairing is not happening then. So the polymerase, so let's say this is not DNA anymore, and the RNA does not have thymine. Instead of thymine, it has uracil, but there's very, very small change in thymine and uracil. So the RNA polymerase comes, and let's say this is not there. So the polymerase will come, and it will first bring in an A. It will read the sequence from this, it will read the sequence from 3' end, synthesizing the mRNA strand from 5' to 3'. So it will go in this direction, and then it will say, oh, now I put in this nucleotide, let me put in a C, then it will read this again, put in a G, and then it will read this and put in a U. So this is the mRNA sequence now. And the other strand of the DNA has basically just separated. So it's not being read. This is the coding strand, that is the non-coding strand, and so this is how you end up, this is the non-coding strand is the template strand for the RNA, and the coding strand is going to be identical to the coding strand of DNA is identical to the mRNA sequence, except for U and T. So that's how it works. So this base pairing concept is actually, this is so easy, and this is really the basis of everything. So now look at this here. A single mutation in a base has occurred in DNA, in 1 gene. And you can see that instead of a mutation has occurred in 1 strand. Instead of GAG, it has become GTG, and the bottom stranded CSE. mRNA becomes GUG, and guess what? Suddenly, this codon, these 3 bases, this sequence codes for valine amino acid instead of glutamine. Lo and behold, you have a different amino acid, you have 1 mutation in DNA, which has changed entirely the properties of your protein. I'm not saying that every mutation will change the protein, but in the certain places, if you end up getting this kind of mutation, you will actually have a major effect of this mutation. Now, how many of you really gathered the importance of BRCA1 and BRCA2 gene that Professor Perry talked about for breast cancer? How many of you are aware of that? Anybody? Just one? Girls, this is something that you should be aware of, and boys, you should be aware of it too, because, it's not that you live a life without women. Mothers and wives and girlfriends and so on. How many of you know that Angelina Jolie, how many of you know the actress Angelina Jolie? Oh, quite a few know. Do you know that she had a double breast mastectomy? Why? She has cancer genes. She carries these BRCA1, BRCA2 genes. And so, yes, and that was the test that she went through to be able to understand that she carries these mutations. Her mother died of ovarian cancer. Her grandmother died of ovarian or breast cancer, and she was at high risk for developing breast cancer and ovarian cancer. As a result, she had her breasts removed and her ovaries removed. So, if somebody knows about these mutations and what that single base mutation can do in the protein that can render or increase the risk for certain cancers, then people can actually make an intervention, take care, and do something about it. So this is the importance that I was trying to tell you of understanding that this is important not just for law, but just as your basic knowledge for yourselves as well. Do not expose yourself to mutagens. Do not think about the environment you live in. If truly the cell phone is those whatever waves they are that are going to create a problem in your cells and create a mutation, then maybe think about it. I don't know if there's enough evidence for it or not. Why is this not moving? Okay. Stuck. Okay, here we go. Okay, so now let us look at, I don't think I'll be able to finish my lecture in time today because there's a lot to go through. But here is where I want to now get into that we have naturally occurring mutations. This is because of our environment. This is also because that's just how biology operates. There are genetic changes that take place, and that is the basis for evolution. That is the basis for selection and evolution and the survival of the fittest, you can say. So these mutations are tested, and only those that are very fit for the environment that they are, those people, those organisms are selected, and then they go on. So those are naturally occurring. Now, we can induce mutations, and Professor Perry talked about them, that what is natural. In the past, people were exposing plants and so forth to gamma radiation and to other mutagens, and then selecting for those. I'm using crops, again, plants as an example, because a lot of biotechnology has happened with plants, okay? So this is my reason for giving you those examples. But we are coming to humans as well very soon. So the idea is that those mutations were created by exposing the organism to mutagens and then selecting for the trait that we wanted, such as high yield or resistance and so on. Oh, sorry, now what happened? Okay, so those are induced mutations. Now, How many of you have heard about this CRISPR-Cas9 technology? Anyone? Major technology, yes, in biotech. So everyone should know, as youth of today, just as you would know any other technological advancement, you must know about this. And then of course transgenic technology, you all know BT cotton and so on. So that part is easy for you to understand. So let us just look at first the very basic DNA recombinant technologies. Okay. And I'm giving you these in the form of cartoons. Think of DNA as a rope or a thread, whatever you'd like to think of. And all you're going to do is use scissors to cut it, and then just use some glue after cutting and doing whatever you want to do with the two ends, glue together something. So scissors and glue is what you need. Well, in terms of biology, they are restriction enzymes that cut DNA and the ligases that join DNA. So these are two proteins that function like enzymes, and this is the job that they do inside the cell. Everybody with me on it or no? Anybody not understanding it? Okay. So now we go and say, what do we mean by recombining or making a new DNA molecule? We take two DNA molecules which are from different origin. Let's say this is from bacteria, and this is, let's say, this here is bacteria, and the brown is human, let's say. So what we've done is we've cut both of them with the restriction enzymes that are scissors that have cut out the DNA, and we bring in this little piece, because this is the one that carries a gene of interest, and we now insert it into this gap that has been created by removing this DNA of the original bacterial DNA and putting in the new DNA in here, which basically is that, and then just closing the gaps between the two DNA by, perhaps you can think of as glue. That's the ligase enzyme does the job. Suddenly what you have is DNA from two sources linked up together and functioning as one. And when we have this, we call it as a recombinant DNA. Okay. So now when we come to genetically modified organisms, we also call them as transgenic, because we are bringing something from another place, or genetically engineered, or genetically modified, however you want to say it, but these are the terms. Now you can ask that the DNA that is, let's say this is the genetically modified organism, in this case it looks like corn to me. This genetically modified corn can have that foreign DNA that has been cut and pasted and brought in again, either from its own self, not necessarily from this plant itself, but from another corn plant. So it's of the corn origin, or of another plant, any other plant. You can think of any other species, cotton or apple or anything. Or it could be from a fish, or it could be from humans. So the idea is, if you have brought in that DNA in a recombined form, introduced it and regenerated the whole plant, suddenly this plant, every cell of this plant is going to contain that foreign DNA. Even if it comes from its own species, it's still foreign because it has been extracted, cut and ligated with another DNA and then put in here. So this is still a genetically modified organism, even if the DNA comes from here. So then the beauty is that how would you, once you've cut and pasted and made a recombinant DNA, how can you introduce it in an organism and regenerate an entirely new organism that is genetically modified, which contains every cell of it has the foreign DNA? Is everybody marveling at it or no? Yes? Okay. So think what scientists do. We look, we observe, we study. They found that on plants, a bacteria was making cancers on the plant. And these cancers, it's called crown gall. These cancers on the plant, when they were studying the biology of it, they found that there is a bacteria called agrobacterium that is hijacking the plant to produce food for the bacterium. Now, do you guys understand cancer? Cancer is a mass of in human body, mass of undifferentiated cells. They just grow uncontrollably. But they do not differentiate into lungs or hearts or anything. It's just a mass of cells. Now, those cells have just escaped control of division and differentiation, so they're just growing, and the cancer is taking over to just grow. Well, in this case, the bacterium is taken over and induce the cancer in plants, because the more the cells that it infects and produce and makes the plant cells to produce food, the more bacteria will be produced. Now here the bacterium is shown with teeth, okay, because what it's doing is it's using the plant cell to make food for itself, and the more the cells, these undifferentiated cells, the more the bacterium grows. So now let's go back to the bacterium. What it does is it transfers a small piece of DNA into the plant genome. The plant DNA, it transfers that, and as you can see, that DNA becomes incorporated in, this is the plant cell, plant nucleus, plant chromosome or plant DNA, and it has become incorporated into the plant DNA, and this is containing genes for producing food for the agrobacterium. Make sense? So it's almost like you going and inhabiting a house and now taking over the house and saying, hey, make food for me, feed me, and I'll live here nicely and comfortably. Same kind of an idea, and I'll grow here. So you have that happening. What scientists did, they said, okay, what if we remove the genes that make food and make for agrobacterium? Instead of making food and making the agrobacterium grow, let's replace those genes with genes of our interest that we want to grow in the plant. And as a result, the DNA will get incorporated that we want, and then we will have a transgenic organism of our own interest. So, they decided to make a recombinant DNA. First they extracted the agrobacterium DNA, removed those genes that make the food for agrobacterium. Instead they put their own genes of interest, and then they use this agrobacterium, put back that DNA here, they use these cells to infect plant cells. Plant cells got infected by this, the piece of DNA got inserted into the chromosome of the plant genome, and this is where you should be asking the question that what happens to the cell then? Guess what? In plants you can regenerate the entire plant from a single cell. Do you think we can regenerate a human from a single cell? Would you like to? Yes? Do we say the person that we like but we can't have them, can we claim this person? and then regenerate. Now I know ethically it would be very this thing, but you never know what the future holds. That's what your science fiction movies show you, right? Okay, so in plants, with a single cell that has been transformed, you can regenerate the entire organism. And lo and behold, every single cell of this plant has the foreign DNA. And all of a sudden you have a genetically modified plant. So in other words, very simplistically, agrobacterium DNA, the genes that make food have been cut, replaced by genes of interest, used agrobacterium as a vector to put this DNA into the plant DNA and to regenerate our genetically modified organism. Okay, now yes. Yes, very, very true. So in stem cells, that's a very good thing, because those are very, very good thing. Because in plants, you can have differentiated cells like leaf cells, and you can transform and de-differentiate and then re-differentiate them into an entire plant. In the stem cells, that the whole idea is this, that if you can find the signals to give to the stem cells to develop into certain kind of tissue cells. So if you want lung, and people are developing, I don't know how many of you know, but they've developed little hearts, little lungs. Of course, they're not fully functional yet. They cannot be used for transplant yet, but they are. They've done it at a very early stages. Those organs have been developed. So indeed, if you wanted to make those cells and transform them, you could do that kind of technology even more precisely in human cells or in animal cells, because there is recombinant homology, and you can target this kind of thing in the stem cells. But then you still have to redifferentiate those stem cells into what you want it to be. Or you can just use the stem cells and target them to, let's say, the spinal cord or whatever, and hope that they would become that. And indeed that is happening now, and I'll show you some examples of the CRISPR technology. So very good, very, very nice. Where I do want to now bring up the question for recombinant DNA technology is that, which Professor Perry will talk about in terms of what law implications or legal implications it has, is that when you actually make a construct, a recombinant DNA construct, it's not as simplistic as I have taught you in just cartoons. You need control sequences to actually monitor the expression of the gene. Then there have to be some kind of selection, so a selectable marker. Then you want your gene of own interest, which comes from some organism or some source. And if you read this, I'm not going to read this for you, but if you see this construct that was made to generate the golden rice, then you can see now that if each of these sequences have been patented by different groups of scientists or corporations or so on, what kind of problem there would be to bring this golden rice to the market. And how many of you understand the scientific significance of golden rice? Anybody? Do you know that there are a lot of people who have deficiency of vitamin A? And the kids who have that, what do you think, what is affected when you don't eat enough vitamin A? Louder? Eyes, exactly. And your eyesight, your eye development, and other things, that's not the only aspect, but there would be other developmental abnormalities as well. So if you have that kind of deficiency in Africa, and if rice is one of the staple foods, well, what is the way to give them vitamin A? Maybe, yes, it's okay to have golden rice for the population. So anyways, this is what, I don't know what the status of golden rice is right now, but this is the kind of different sequences that have been used in making this construct. How are we doing for time, Saurabh?
So, we are crossed.
We've crossed the time, so
We need to continue this, we can do that tomorrow.
I think we should, because I'm beginning to see that students are getting a little bit in this heat, a little bit tired.
We can continue this tomorrow also. If you think that we can break at this moment.
Yeah, I can. Are you guys okay? Do you want to continue? Yes or no? No? No, not today, tomorrow. Yes? Okay. Do you find it interesting? Okay, great. So let's do this tomorrow. I know it is really hot and humid and you're getting tired, so let's do this tomorrow. Thank you for your attention.
Also,
Just finished.
tomorrow we'll start 15 minutes early, okay, so it's not 2:30, it's 2:15, right? So because it seems that 5 o'clock, for some of you, there is some other lecture also scheduled, I don't know, so told me that so we'll start at 2:15, so please be there at 2:15. So I have 3 names from outside, I don't see those people, so I mean, Poonam, Devendra, Devendra, in case I'm missing out. So okay, it's 2013 first, right? Roll number 1, 2, 3, sorry 6, 14,
Yes, sir.
15,
Yes, sir.
17, 18, 23, 26,
Yes, sir.
27,
Yes, sir.
30, 32, 38, 39,
38.
39,
Yes, sir.
40,
Yes, sir.
41,
Yes, sir.
43, 44, 45,
Yes,